First of all, I would like to thank Sukan and the NTSAD for giving us the opportunity to introduce Azafaros, because this is the first time I think we talk at the conference, and to also discuss our main project in GM1 and GM2 gangliosidosis. We just got actually the T-shirts this afternoon, so I could put it on today for the conference. So this is very nice. Thank you so much. Just to say a few words about Azafaros. Azafaros is a strongly science-driven company. We ambition to find and develop innovative disease-modifying therapies to tangibly change and modify the clinical course of rare metabolic disorders. So to put this in the context of today's conference, I should also say that we value enormously the central role of patients and parents and families when it comes to developing new therapies. And indeed, we like to engage with the clinical experts in the field with all our passion. So our primary focus at Azafaros is on lysosomal storage disorders, and specifically the GM1 and the GM2 gangliosidosis, which is indeed the topic today. For this, we are developing a small molecule named AZ3102, which can be administered in the mouse, and which has, we believe, potential as a disease-modifying therapy. In addition, we would like to expand to other rare metabolic diseases and other therapies. And for this, we have embarked on a new drug discovery program, which I will not talk about today, but which is also quite interesting and important to us. So what about GM1 and GM2 gangliosidosis? I think everybody is quite familiar with information on the genetics, the epidemiology, and the medical condition of patients suffering from GM1 and GM2 gangliosidosis. And most importantly, we have recognized the vastly unmet medical need of these two diseases and the potential of the small molecule AZ3102 for these diseases. What is really critical is that because the two diseases share important pathogenic and clinical features, we plan to address both diseases within the same clinical development program. And I will, in a minute, elaborate a little bit on how we would like to do that. This is a rather complex but important slide about AZ3102, and please bear with me. So AZ3102 interferes with two key pharmacological targets with similarly high potency. These enzymes that we are targeting have a central role in glycolipid metabolism. And this makes AZ3102 quite unique, we believe. The non-clinical data that we have generated over the last 2 years have shown that the brain is exposed with the molecule, and we also have generated evidence that the pharmacodynamic effect is indeed taking place in the brain. On one hand, AZ3102 targets the enzyme glucosylceramide synthase to reduce the accumulation of GM1 or GM2 gangliosidosis, depending on the disease. This modality is known as substrate reduction therapy. On the other hand, AZ3102 inhibits the enzyme non-lysosomal glucosylceramidase, named GBA2, with the goal to reduce the intracellular level of other harmful metabolites and to ameliorate the impaired lysosomal function. The absence of inhibition of intestinal digestive enzyme with our compound is also clinically relevant, and unlike predecessor compound, we do not expect unwanted gastrointestinal side effect in humans. And this has now been very well documented in our non-clinical studies. The pharmacokinetics of AZ3102 allows for a once daily administration. And finally, we are planning to develop a pediatric formulation especially designed for children with the diseases GM1 and GM2 gangliosidosis. So where are we? We have recently announced the start of dosing with AZ3102 in healthy human subjects. This is in a two-part phase 1 clinical study, single and multiple ascending dose. This study is actually taking place in Adelaide in Australia, where COVID has much less impact than in other places in the world. Importantly, we have decided to measure drug levels, not only in plasma and urine, as it is usually done, but also in the cerebrospinal fluid. In addition, we are measuring pharmacodynamic markers, both in plasma and in the cerebrospinal fluid. The results of the study are due in the second half of 2021, this year, and will help us define the dose levels and dosing regimen suitable for administration to patients in future clinical studies. So today, I am very proud to announce that this summer 2021, we will initiate recruitment in a dedicated and fully prospective multi-regional natural history study. The study will be conducted in patients with late infantile and juvenile onset of the two diseases, GM1 and GM2. The study will help design the future pivotal study with AZ3102 by defining the longitudinal trajectory of the major neurological disease aspects. We want to build a solid database which will serve as external control group for the pivotal study, assuming that we will not want to have a placebo arm in the pivotal study. We also want to identify factors associated with different risk of rate of disease progression. I think what can be of real interest for our audience today is that we are designing the study as such that parents can be directly involved. To this end, we are developing an app to allow parents to complete validated questionnaires and patients' diaries from their own laptop or smartphone. To facilitate study participation and minimize the burden of traveling, especially these days, to study site, we will implement virtual assessment with a secured investigator-patient video interface. So as you see, we are really trying to build into the study technology to facilitate reduce the impact on patients and their families. Clearly, the natural history study will be an opportunity to build an international community of families and clinical sites which are willing to engage in the project with a long-term perspective. This study will be multi-regional, involving up to 35 sites in up to 12 countries, major countries. We plan to enroll at least 75 patients with age comprised between 2 and 20 years, and with an onset of neurological disease after the first birthday. We have identified a digital endpoint based on a wearable sensor to monitor gait, balance, and other ambulation aspects. We have already started reaching out to sites, and we will keep doing it in the upcoming weeks. In the last few months, we also have been able to contribute to very interesting initiatives from patient advocacy groups, which you can see here. This project will also help us and the scientific committee design better clinical studies. Now, in the interest of time, I will not detail this, but we certainly hope to do much more in the future. All these cannot be done without people. Azafaros was able to bring together a team of very experienced and passionate individuals whom you can see on this slide. We come from different horizons and countries and diverse backgrounds, and we have all been working in the field of lysosomal storage disorders in one way or another in previous positions. This is a great team indeed. So I'd like to conclude and sum up. Azafaros is focusing on rare metabolic disorders for which the unmet medical need is high. We concentrate on small molecule therapies, which is our expertise, and want to apply the best research to our programs. We are developing AZ3102, a small molecule, as a unique dual glucosyl ceramide modulator, now at clinical stage. We're about to start a prospective natural history study. And most importantly, we clearly see that involving patients and parents is key to the success of innovative clinical research, and in particular, what we're doing now in GM1 and GM2 gangliosidosis. I would like again to thank you very much for having us today and for your attention. It's greatly appreciated, and I will be happy to take questions.
Thank you very much, Dr. Moran. That was fantastic. One question that I have, and I don't see any questions in the Q&A yet, but one question that I have, and I think you addressed it, is the potential for off-target effects or intestinal toxicity with this compound versus other compounds that have sort of the same mode of action.
Well, I can clarify this. We have already actually communicated this. The compound we're dealing with is what we call an L-Edo analog, or L-Edo as a sugar, and these L-Edo as a sugars do not carry any liability as inhibitors of the intestinal sucrase or maltase or lactase. And this is unlike a drug that we briefly heard about today, which is miglustat. And all the non-clinical studies that we have performed with this compound, and we have done many, we do not see any of this in those studies.
That's very good news, and I know a lot of the families will be glad to hear that.
I have to say it was designed from the beginning. Let's say, we selected the compound for a number on a number of criteria, and that was one of them, obviously.
Great. Very encouraging. Any other questions from our panelists or attendees, families? Okay, if not, thank you very much, Dr. Moran. It was fantastic. Thank you for being here.
Thank you. Thank you very much.